Well, I think I just started the recording here, so we'll be able to. I'm gonna probably put this up on the. I got a new YouTube account now. I think all my students have been like encouraging me to find some way to make this accessible, given everybody's schedules, and I can easily do that. So I think before we end, I'm getting pretty good at reminding myself, but please remind me before I end this thing that I send you a link and you guys are able to sign up on the attendance forms. That's kind of a big deal. I want to make sure you guys get your attendance. So, but that'll probably be closer to the end. We'll have to see how long we go today. If we got a lot of questions, we'll definitely address those. The quiz, is the quiz up yet? I think the quiz comes open on Monday, right? It's not available yet, right? You all haven't seen it? Okay. Okay. Let's make sure we know the baby. Make sure we know the baby. Make sure we know the shoulders of the baby, the cheek region of the baby, the neck region of the baby. If there's any students here that have me for lecture, or excuse me, for lab, does anybody here have me for lab? Okay. Okay. If you have me in person, you probably listen to me harp on the quadrants. Yeah. And that's going to come home. That concept's coming home on the quiz. So, and I knew it would. I knew it would after I looked at the quiz. So quadrants, I think you're going to want to take a look at that. Both, Karen, both. I'm trying to think what else I saw on there. The babies, the babies just take so much time. So it's so detailed. That's why I wanted to point out a lot of the other stuff. There's like maybe 3 conversions. So don't overlook the conversions. And then a lot of the other stuff, if you think about when you read these questions, you have to make sure you know what they are asking, and just don't assume. Read the question. I know you're timed. I know this kind of like gives everybody anxiety because it's timed, but if you read the question and you study the material, then you know what it's asking. Okay, you know what it's asking, then you can answer accordingly. So, and don't be fooled by the easy give me answer. Okay. So if we ask something about, if we ask a question about what is the, what if the hair, what system is the hair part of, I guess is what I'm trying to say. Okay. And if one of the answers is follicle system, well, there's no such thing as follicle system. Okay. Okay. Yeah, that's right, Karen. You remember that. Everybody see the chat, what Karen said? So stuff like that. Don't be fooled by the give me answer, because the chances are it's not going to be that easy of a class where we just give you that. I think so, Caitlin. Everybody has their own liberty of basically what it is that the program director will put out the suggested quiz. Okay. Now, most of us will take her suggested quiz. However, some people like to put their own twist on things. So the exams will be exactly the same, but the quizzes sometimes may be a little different. But in all likelihood, that all quizzes will probably be the same. So FYI. So it's 20 points, 10 minutes. So you need to budget your time. Okay, cool. I'm not sure about Morelli. I don't think I've ever introduced myself to him or her. But the quiz will be 20 points, so it's a 20-point quiz. Remember, we have 2 of those in the semester. These are the ones that can't be dropped. So you want to make sure you do well, and you want to make sure you don't get bogged down on an answer. Okay? You do not want to get, you don't want to sit there and spin on a question for 90 seconds or 2 minutes unless it's like your last one. You need to be able to skip it and come back. What you really don't want to do is leave 3 or 4 blank at the end because you didn't budget your time. And this is all part of test taking skills. This are skills that you have to develop over your academic career, but you probably want to start developing it sooner rather than later, because if you're not, you're going to get taken to the bank on poor quiz grades. Poor quiz grades add up, and they really don't help you in the end. I think the lecture will be something completely different, Karen. I think the lecture quiz is going to be something completely different. I'm only speaking about the lab quiz. Since I'm a lab instructor, I know mostly about, excuse me, lab quizzes. Let's see. Quizzes will not be proctored. Only exams are. Quizzes are going to be timed, and exams will be timed too. But the quizzes are timed to ensure that students just don't go burn through their notes. You can, if you want to try to cheat, you can try to cheat. But the way the time limit is, I'm not sure if it'll do you any favors, because you'll be flipping through your notes and you're going to be sucking up so much time that it will not be beneficial. And cheating's obviously not allowed, but it's just no way for us to police it through the quizzes. So, okay. So I think just make sure you touch on everything. The skin layers is another one I noticed. You want to start the skin layers and go from there. So I think that gives you all enough hint on what's going on there to get a good first step on the quiz. Okay, so Lab 2. I'm sure we're not going to be too impressed with Lab 2, just because we're worried about the quiz. And I think it opens up tonight at midnight, by the way, and it closes Thursday at midnight. So you have 4 days to get it done. And this one can't be dropped because it's a 20-point quiz. So keep that in mind. Okay, so now I am pulling up my Lab 2. Is there any other questions, by the way? I know some folks like to talk in front of their microphone, and some folks like to chat. So we good on? Okay. And is this a lot of people's first spotlight session? Cool. Okay. Yes, that's correct. This week's the first one. Okay. So we've had, okay. So a lot of people, this is their first ones. So obviously you haven't had me if this is your first one. And I think at this point everybody kind of understands the syllabus. You don't want to miss more than 3 quizzes. Keep that in mind. You don't want to miss any more than 3 quizzes or lab quizzes. I'm speaking about lab quizzes. I'm sure lecture quizzes are the same. Or if you miss more than 3 lab quizzes, you fail the lab, which means you fail the whole class, and you don't want to do that. Okay, so that's an important point. All this can basically be found in the syllabus. If you have any questions about the syllabus, please let me know. You want to make sure you get attendance for at least, 2 or 3 of these before the midterm. I kind of recommend people coming to a fairly good amount of them, because especially if you come and find me and it's coming up on quiz or exam time, I'm going to try to steer you in the right direction. So at least, because sometimes there's so much material, students will get bogged down on some low yield material, and I'm going to try to kind of direct you into the high yield material. That way you spend your time wisely when it comes to studying. So I think that'll be kind of beneficial if students were to sign up for these spotlights more often. And of course, the benefit is you may get the drop on the quiz information or a drop on the exam material. And that's kind of what these spotlight sessions are used for, is kind of supplemental information. So, okay. So let's see. I talked about test taking skills, talked about the syllabus. I think I've beat that horse dead on excused absences. I think we all kind of know there that you want to be able to show up, and you better, to avoid any confrontation or misunderstanding or chance of losing points, you just kind of want to be upfront with your lecturer or lab person if you were to miss a quiz or an exam and you think it is an excused absence. That would be like a religious holiday, a university trip, or the most used one, is, you know, either yourself getting sick or an immediate family member is in serious illness, or you know, perhaps even up to death. So I think a lot of us are pretty lenient on that, and we're understanding. When I went through graduate school, I think my grandfather passed away right in the middle of the exam block, and you know, I had to work with my professors, and they were very nice and just trying to accommodate me because you know, I think his funeral was the morning of the exam. So, and we're all pretty lenient on that. You just don't want to make a pattern of it and raise eyebrows, and it'll prompt more questions. So makes everything kind of uneasy. Okay, so Lab 1 was basically like a introductory tour of everything that we're going to look into later in the semester. And if you didn't have me for lab and you want to, I think, did everybody get the lab key 1? If you didn't and you want it, I know my folks were given it to help study. But if you want the lab key, lab 1 key, I guess is what I'm trying to say, you might want to email me and I'll send it over. That way you have the proper material to study. Maybe you already got it from your lab instructors, but I'm just going to throw that out there. Okay, and tutoring. Tutoring is, let me see if I can share my screen. Yeah, email me, Caitlin. This is my email. All right, just drop me something and I can get it to you. It goes for everybody else too. Okay, now let me see. I want to pull up the tutoring real quick because this is a big deal, and I'm going to try to share my screen, so bear with me. Okay. Let me click it up here, here. Okay. Everybody should be able to see my screen. So Ellen Sam is going to be our Bio 137 tutor, and she's going to tutor on Sundays from 1 to 4, or you can do by appointment. And this is her email. So if you want to try to get it later, you're more than welcome to reach out, or you can probably find it either on the tutoring center or somewhere else. I think it's in the syllabus. There's some tutoring information. So, all right, I'm back. So I think that'll be good. You know, sometimes they say things a lot better than myself or other instructors, and it just makes sense because, you know, you get another pass at the material. So, and that leads me to another point. If you're trying to memorize some stuff, the thing about Lab 1 is that you can probably memorize stuff and get away with it. When we get later in the semester, it'll be a lot harder to memorize things. But this first lab is kind of like memorization. But you want to spend, a lot of times students will complain that, yes, they knew when they got to their house and they knew it when they were studying the library, but then when they came to the quiz, they blanked out. And that probably means that you probably did not know it as well as you thought you knew it. Or you studied it and you stared at it, but you never tested yourself over it. That's the big thing. They'll sit there and they'll do the flashcards, or they'll, you know, but if you write it down, I always wrote things down. Like I wrote it out. That's how I learned how to learn the material. I wrote it out. And then I would test myself, you know, and then I would sit there and have a conversation with myself and make sure that I understood, and that when time came to take the quiz or the exam, I didn't blank out. I'm just sitting there spacing out an answer, and then before you know it, 50 seconds has gone by and I'm still confused. So it's just something, it's another test taking skill that you need to kind of start working in, because eventually you're going to take licensure exams and they're all going to be timed, and they're going to be having these questions that are worded where you should be expected to know what the elementary language is. Like we talked about anatomical position, right? And we said something is posterior, anterior, superior, or inferior. Well, the more time you spend with that, questions will be posed assuming that you know the definitions of superior, anterior, posterior. So that's kind of why it's important to lay the foundation now in Lab 1, so you can build upon it later on in the lab and your academic careers. And another thing we're going to build upon in lab 2 is microscopy. Now, I know some of you guys probably, if you have me or other instructors, you're probably on the online section. However, if you are in person and you want to come to, well, at least with my people, I do Zoom meetings and in person. If you come in person, you can get a chance to work with the microscopes. So that's what we're going to be doing this week. And the cool thing is that I'm a third shifter and I work in the clinical lab at night. So that's pretty much all I do is playing around on microscopes. So I've gotten pretty good at getting maneuver my way around these microscopes, and they're kind of cool when you can get things focused in. I think it really shines a light bulb on with students and what they're looking at. And some of it's more complicated than others because some of it's tissues, so you have to really study the characteristics of the tissues. I think we're probably getting into that lab 3 or lab 4 if they haven't jumbled everything around, and that can be more of a demanding lab. But we'll get to that point, when we do. Let's just learn how to work our way around the microscope and learn the different vocabulary terms of the microscope. So let me pull up lab 2 here, and let's see. I'm going to scroll down to page 2.2. So all these different vocabulary words are words that we're going to want to need to know. Now at the very top, the oculars. The oculars are also under magnification. And when I mean magnification, it's like looking for a magnifying glass. So when we have the ocular lenses are going to be under a 10x magnification. So if you just want to write 10x ocular lenses, please do. Now, I want to direct your attention down here to the objective lenses, which is like those little cone shapes. Let me see if I can share my screen. My students tell me that I need to start sharing my screen so they'll listen to me like a talking head. And I think you can see my mouse. So here's your ocular lens right here, right? So this is going to be 10x right here. Now these are the objective lenses, and this is like on a rotating. You can pretty much take your finger and spin this around. And these objectives are of different magnifications. The yellow, I think maybe like, what is it, 10x. The blue, I'm going to call this one blue. This one would be like, you don't have to know these, but I'm just giving you examples. Like this would be a 40x. Then you'd have another objective of a 50x. And then if you have a really good microscope, you'd have another objective of 100x, right? The higher the magnification, the smaller things can appear on a microscope, okay? Like for instance, like if we're talking about the molecular world, a parasite is huge. Like a parasite, if you were to see like a parasite, it would be like really large. So you wouldn't need a large magnification. You could see a parasite at 5 or 10x. Let's see, another large cell, sperm cells are considerably large. Epithelial cells are large. Now, if you wanted to get in and look at like the different organelles of the cell, let's say like nucleus or nucleoli, or you won't be able to see like mitochondria and stuff like that, because the reason how we could see these different organelles is that we've stained the cells. We've stained them with with these chemicals that help kind of bring color contrast to different parts of the cell. The membrane would be stained, and then the nucleus would be stained, and then the nucleoli would be stained. Now, for a virus, like viruses are like the smallest of the smallest. So you would need almost like an ultra large magnification objective to see it. These are just regular microscopes. You would probably need what's called an electron microscope. Those are the best that the best science can offer. And, they can run anywhere from $500,000 to $1 million, probably more than $1 million. So like if you're looking at COVID-19 and you're looking to see what the features of COVID-19 is, you're going to need like a very high magnification microscope. You know, something like a bacteriophage, that is a virus that infects bacteria only. Those are a little bit bigger in the virus world. You can kind of see those with like what's called a confocal microscope, still a very expensive microscope, maybe about $100,000, $200,000 microscope. These are your run-of-the-mill, you know, still pretty good, but I use something like this in the hospital. You know, we use something that's pretty much a very, yes, it's highly calibrated, and but it's still gonna have a pretty high objectives on it. We would use these for probably detailing what cancer cells look like, or looking at some kind of tissue histology for any kind of malignant formations. What else? We do sperm counts. You do sperm counts on them. But I think up until there, that's really about it. You're probably looking at basically you have an objectives enough on there to where you're looking at the different features of an organelle of a cell, not organelles of the cell. Excuse me. Different features of the cell. Like you can probably, if I can find a B cell, I can probably tell whether it's pumping out antibodies or not, just by how the form is, the shape of the cell. You can also pick out like what bone marrow cells look like versus regular cells. So that's just kind of a quick introduction to what these microscopes can do. So we need to cover all the different vocabulary terms of the microscope. And unfortunately for me, there's really no cool stories to add to these other than just learning flashcards. And as you can see on page 2.3, let me see if I can, I'm going to bring you guys back here. I'm sorry, I need to get used to this. So on page 2.3, we have the part and the function of it. So spend some time on this. I want to highlight the condenser, okay? The condenser is something that's interesting. It's kind of, it's this part, well, this part right here, and that's actually a whole device underneath the stage that can move up and down, and that will allow it allows certain, I guess, certain bits of light to come through, certain variations of light to shine through, and we will use that for cells that may be transparent. Like epithelial cells are very light, and you can easily like bleach them out with too much light, where you think you're looking at something and it'll be loaded down with epithelials, but you're having too much light come through. It's kind of like looking straight, like for us, it'd be like looking straight at the sun, and all of a sudden you can't see anything because the light's just bleached out everything, and it's like it's all white. But if you were to turn down the condenser, then you will be able to kind of provide less light to enter into the specimen, and all of a sudden it's like, you know, we just flipped on the lights and we see all these epithelial cells. It's kind of hard to explain unless you do it in lab. It obviously, with these different semesters, you know, chances are you'll probably, if you don't get your hands on a microscope now, you'll definitely be using them in microbiology. So don't worry about it right now. Okay, so we have coarse focus and fine focus. That's just how it sounds, right? I think now I want you to highlight something. You never use the coarse focus on high power. That will probably be something that shows up in a quiz question, I think, in past years. So make sure you highlight never use the coarse focus and high power, and I think there may be something with the fine focus too. So, you know, when we are under high magnification, we do not want to use a lot of movement with the focus. We want to use fine movement, because basically what we're doing, let me see if I can get myself back on here. No, maybe I'm too slow. Okay, I think you guys can see me. So when we look through a microscope, think like this is the microscope, right? And this is our specimen. When we are focusing, we're basically focusing at different planes of that specimen, okay? Everything's 3D, right? It doesn't look 3D, but in the molecular world, it still sits off the ground. And when we use a microscope and we focus, we are focusing from the top and then we turn it, and then the stage will move, and then we are focusing at different layers of that specimen and see what's going on, because it's just not one plane. It's like it's a 3D plane. So when we focus, we're using the fine focus, and we're looking at different planes of that specimen. All right, it's kind of like looking down in the water. Like, let's say you're looking down, you're underwater, and you want to like swim deeper into the water, and so you can see different layers of the ocean or something like that. Different depths, I guess, is what I'm trying to say. Maybe different depths of the ocean or something like that, and see what kind of organisms are around. It's like taking a submarine. The farther you go down, the more cooler things you see. Same thing with a microscope. As we focus, we're focusing on different layers of it. So I'm just trying to explain that as best I can. Okay, the objectives. Let's focus down. Let me see if I can bring you guys back to my screen. Hold on, I got a question. Let me hold on. I just saw it. It just went away. I'm sorry. So cells could be under others. Yes, you bet they can. Yes, and some cells on top may not, may look normal, and then you start focusing lower, and these cells may look more malignant. If you think about it, when cancer invades tissues, all of a sudden, like tissues, like you can always tell when cancer's in tissues, because normal tissue has like really like everything's like layered, like it's kind of like, think of your thumb. If you look at the ridges of your thumb, right, what makes up your fingerprint, everything seems to be fairly in line with each other. It's all it's got this continuity to it. If cancer is involved, all of a sudden you lose this continuity, you lose this layering, this nice fine layering. It's called I forget what it's called, but you can see like an invasion, so to speak, like something's been pushed aside, or something's growing out that is kind of like disrupted the landscape of the tissue. And basically how doctors and surgeons figure out, like, let's say they're extracting a tumor or some malignant tissue, they will cut around it, and then they will send it to the lab, and a pathologist will look at it. And depending on how the tissue looks will really depend if they got it all or if they didn't. Okay, that's how they really determine if they think they got it all or not. You want to make sure that you take all the tumor plus a little bit of good tissue around it, because it's kind of invading. Yeah, it's kind of like it could be like a frozen section. I'm not too familiar with histology, but obviously like a histology tech would be able to answer these questions. Yeah, but when you're taking a slice, like a frozen section or a frozen slice, and you're sending it to the lab, you're going to want to make sure you take the malignant piece that is causing this abnormality, as well as healthy tissue around it. Because if you have healthy tissue around it, then that means that chances are you've gotten it all. So that's what's kind of interesting. Yeah, it's margins. I think that's the word. Yeah, Karen, I think you're right. It's called margins. Good margins. Yeah. That's the word I think I've heard other people talk about, which is kind of interesting. Okay, so let's see. I brought you guys back to me. Objectives, you have low power and you have high power, and the oil immersion is always going to be under 100x. So I want you to star that. You're going to go over this more when you get into labs, but this is always kind of interesting because these topics show up. What is low power? What is high power? And when do we use oil immersion? Okay. And then, like I talked about earlier with the ocular lenses, those are going to be 10x. So if a question were to ask you, what is the total magnification? If you have a 40x objective on a specimen and you're looking through it on a microscope, what's the total magnification? All right. Now I'm going to see if you all know that answer. It's not 40x. It's too easy, right? That'll be an option. That'll be an option though. But there's going to be a multiple choice answer that says 40x. But unfortunately, that's the wrong answer. Does anybody know the true answer? What is the total magnification? Remember how I told you about the oculars? And the oculars have magnification? Somebody chat me something before we can go on. I want you all to kind of think about this. Well, you're close, but so it'd be, so it's 10x, right? So what's 10 times 40? Yeah, it's 400. You're right. It's 400. So that'll be the total magnification. So I want to make sure you guys understand that. If you have low power and it's just 4x, all right, so 4x is the objective, and then the ocular is 10x. Oculars are always going to be 10x. So it's going to be 4 times 10x, and the total is 40x. There's exercises in the lab for this. So, I don't want you all to get all uneasy with me or on me. You're going to be practicing this in lab this week. Okay. And I think that's kind of all the interesting parts of the microscope that I want to have you circle or star. There are, let's see, there are a procedure, I think is what I'm going to call it, on 2.4 and 2.5. So you have the paramecium. This is something that you can do at home or you're going to do with your lab instructor. It'll talk about like scanning. Like a scanning is also called like low power. You're kind of like seeing it from like a bird's eye. We're not really like getting down and dirty with it. We're just looking at it from a very high standpoint where you can kind of see the whole field, right? There's no reason to like have a super high objective on there if you're still looking for your target. You only want to switch to high power when you found your target and you want to like zero in on it. A 4x or a scanning X is more like looking at it from a bird's eye view, very high up. Okay. Let's see. We're not going to go over estimating size of the field. We're not going to touch on that. The lens effect. So everything's upside down. That's what that means. Everything is upside down with the lens effect. That's kind of how microscopes work. So you'll see this when you do it. When you put the letter E, it's going to look upside down, which is, it's just something it's kind of like a mirror image. The mirrors in the microscope flip and reverse the image before it reaches your eye. This is called the lens effect. So it's when these bolded words come up in the lab, these bolded words there's a high likelihood they're going to make the quiz. Okay. If something is underlined or if there's a little bit extra attention made towards it, you can definitely star that or highlight it. And there's, it's a concept that you don't want to skip over. Okay. There's a thread. The depth of the field is kind of like what I was talking about with the focus. I didn't use that term depth of the field, but this is on page 2.9. Let me bring this up for you guys real quick. Right here, this is the depth of field. This is something that I was kind of talking about earlier, how we have different layering of the cells, and when we focus, we are focusing through the field. We're focusing up and down through the field. So, and then the next page, this is like the exercise I was going to talk to you about with the magnifications, and we'll save that for when you do that in lab. And now the last part is more like a wet mount, and that's kind of like that's what we'll do is when we come to Tara. You don't have to. I don't require my students to print the lab out, because basically all lab, I mean, if you haven't noticed already, lab is pretty much quizzes and exams, right? So if you want to print it out and study it on your own, then that's fine. I mean, that's how I got through all my classes, that I had to write things out, and the more I wrote things down, the better I understood it. But you're not required, at least for me, to print it out. Because it's, lab is only quizzes and exams, so we don't turn anything in. Okay, I think I answered your question there. All right, so wet mount at the end of the lab, at least for in-person lab meetings, we're going to have a cheek swab, and you're just going to you're going to take a toothpick and try to get some cheek cells onto a slide, and we're going to stain it, and yeah. Yeah, I agree. Yes, I agree, Caitlin. That's what works for me. I know everybody's different, but um, you know, that's what worked for me. Um, and then you can stain them, and basically we're just looking at a bunch of big epithelial cells. That's what your cheek cells are. It's just epithelial cells. So, all right. So I want to scroll down to cell structure. Cell structure is also, when it comes down to more memorization. That is something that you guys will have to just spend some time on. This is on page 212, and learning all the different organelles of the cell. This may be from high school. Some of you all are fresh out of high school. Some of y'all it's been spent some time since you've been in high school, and that's all right. But this will be just like a nice refresher on page 212. That certain things love to show up on the quizzes and exams, and frankly they're easy points. Yeah, same here. Same here when I went back. So let's see. There's nothing interesting to add here. It's just something you want to spend some time on. I think centrioles are a big one. Yeah, Caitlin, you all probably went over this in high school. So it'll kind of come back. But I think centrioles is something that we want to make sure we know. And number 9, I think, is what mitochondria. No, that's Golgi. Okay, I was going to say I didn't look back mitochondria. Number 9 is Golgi apparatus. So I think those are some ones that I've seen that get picked out, but don't quote me on that. We want to make sure we understand the difference between the rough endoplasmic reticulum and the smooth endoplasmic reticulum. All right, those are part of protein packaging. Smooth endoplasmic reticulum is responsible for like more steroid hormones or steroid proteins, and then rough is more like water soluble. It's getting a little too complicated, but you'll get more into that when you take upper level cell biologies. I don't think so, Africa. I don't. I think you could probably see the nucleus. Now you'll be able to see the nucleus, and you may be able to see number 4, which is the nucleolus. Those are probably about the only time that I can really see like cell structures. The nucleolus will be, if it's a young cell, it should be in the bone marrow, but somehow it's made it out. Because all right, you can think about this. All right, they draw your blood, right? My specialty in the clinical lab is hematology oncology, blood cancers of the blood. So I am kind of I'm not going to say the go-to guy, but I'm one of the guys that they go to for leukemia and lymphomas. Okay, so when I see cells, when somebody gets their blood drawn and they come to the ER and they draw their blood, and I see cells that should be in the bone marrow that are now out in the peripheral, that's usually what that means. When cancer's kind of overtaken, you get a lot of these bone marrow cells that are moved out of the bone marrow or are now circulating into the what we call the peripheral blood, which is the periphery of your blood. Then I can kind of detail where they are in their immature stage. And how we do that, we look at their nucleus and we look at their nucleoli, nucleolus. And we can kind of tell what they're doing and how bad or nasty they look based on the malignancy. That's probably about the only time you could see organelles underneath the microscope. So it's kind of cool. I'm not sure if I can bring some in I don't know if I can or not. If I come across something, I can bring it in. Okay, good. That makes sense. All right, cool. Everything else really not much. Now, if it's a lymphocyte, which is a B cell, and this person has, let's say COVID-19, or they have some kind of viral infection, and they're pumping out a bunch of antibodies, then I can pretty much tell that this cell is what I call reactive, and it is actively producing antibodies. So that's kind of cool too. The cytoplasm looks like a big tongue. You have how you have like the nucleus in the middle, and it's like looks like a little penny or a quarter, like nice and round and circular. If it's reactive, it looks more like an oval. It's like an oval shape, and it's like really, the cytoplasm is like really large and darkly stained, and that means that it's turning out antibodies like you wouldn't believe. So that means it's just you're under infection, and that's what white cells are supposed to do. So yeah, that's kind of interesting too. Okay, so we will talk about. Okay, I guess we're going to do some tissues. We're going to talk about what sperm cells look like, human blood cells, and cardiac muscle cells. We will all get into that in the lab. But if you are taking this online and you won't see this in person, you can always just Google image search these. These are standard cell types, okay? Simple squamous epithelium cells. You can type that in. They're all going to look the same. And I probably encourage you to do that because there's going to be pictures on the quiz or on the exam, and they're going to ask you, what is this? Name this cell. And like cardiac muscle cells are going to be like more rectangular, okay? They're going to be like kind of pinched off at the ends, and because they're all kind of connected to each other, which is interesting. That's how we get if we get into cardiac rhythms, with how they shock people, all right? That's what they're trying to do. If your heart stops, they shock you, right? And that shock will travel through these muscle cells, which creates the heart to beat again, right? These cells are responsible for carrying that electric shock through the cells to create the heart to beat again, okay? When you're normal, you have your own shock, right? You don't need somebody else to shock your heart. If you're have normal, what we call normal sinus rhythm, then your heart does it's shocking itself. But these cardiac cells help that electrical signal conduct through the cells. So you're going to kind of see this like structure. You're going to see this like structure explains function. And what I mean by that is like you can look at the structure of the cell and then understand the function of the cell. All right. Like epithelials are more square-like. They're kind of like wall tissues off. Okay, they may protect it from infection, or they're just there to sacrifice themselves. Like simple squamous epithelium cells. That's like what your skin is. It's like dead skin on top, and basically your skin's there as a barrier and does all kinds of different things. But you can kind of understand that it creates like a wall of protection, whereas cardiac muscle cells carry a conduction, and you'll be able to see this once you get eyes on it. Human blood cells, if you think about it, they look like a donut, right? If you ever looked at a human red blood cell, they look like a donut. And in the middle of these donuts is a very thin membrane. Well, that's where the oxygen goes, all right? If red blood cells, and by the way, does anybody know something cool about red blood cells? If you've had me in person, I wore this out like the first week. Does anybody know anything cool about red blood cells? Either you can shout it out to me or chat me something. I'm going to give you all a minute to think about this too. Okay, nothing. All right, so the interesting thing about red blood cells, when it's a mature red blood cell, they do not have a nucleus. It's one of the only cells in the body that does not have a nucleus. If it did, it couldn't carry oxygen. At one point in their life stages, they do have a nucleus. Babies have them a lot, especially if they're premature. Premature babies have a nucleus, and then eventually the nucleus will get pulled out of the cell because we need to make room for it to make oxygen. Okay, so that's kind of interesting. You can Google what a red blood cell looks like, and you're going to see you're going to see what I'm talking about. If you want to Google what a nucleated red blood cell is, you'll be able to see a darkly stained circle within a circle, and that is the nucleus inside the red blood cell. All right, and then sperm cells. Okay, sperm cells. We know what sperm cells do. They carry all the male genetic material to meet the egg and produce a zygote. Okay, and if you think about it, sperm cells, they have their head at the top, and then they have a tail, and then they use ATP or energy to spin that tail to swim up the vaginal canal and try to find the egg somewhere up in the fallopian tubes, right? And so they have to swim a pretty long way. If you think about that, that's like a big distance. And the one thing, does anybody know like the powerhouse of the cell? I'm sure that's what everybody uses. You high school people should know this. The powerhouse of the cell. Like what organelle in the cell is, yeah, that's right, Caitlin, the mitochondria. Yeah, there you guys go, Lakeisha. Samhama. I'm sorry, I just botched your name. I apologize. You'll probably correct me later on. That's the thing with these. I just like, it's all recorded. And let's see, Africa. So yeah, mitochondria. Anyways, so sperm cells have to travel a long way away to find the egg. They use this tail as motility. Probably not. I don't think they would. The white blood cells would see them as normal, but I'll get back to that, Caitlin. Let me finish my point. The sperm cells take a lot of time to travel to the fallopian tube. They use this tail to spin, which uses energy, right? Where does this energy come from? Well, you just answered it. It comes from the mitochondria. Sperm cells have a tremendous amount of mitochondria compared to all different cell types. If you were to like look through a sperm cell, you'll see damn things loaded down with all this mitochondria because it needs all this energy to rotate the motility so it can propel its way against the flow or against the grain, so to speak, because usually in gravity too, for that matter. So it can find an egg and fertilize it. So that's what's kind of interesting is where you start to see the structure. It really starts to make sense of the function of the cell. Now, to get back to Caitlin's question, does white blood cells attack sperms in the canal? Typically, no. Typically not. I don't think there would be a lot of white cells in the vaginal canal unless you have something like a vaginosis. If you have yeast infection, yes. If it could, like let's say, now this would never happen, but let's say most of the time white blood cells hang out in circulation, right? Now we talked about, I talked about some other class. Certain white blood cells are able, neutrophils and monocytes, you don't have to know this, I'm just telling you, most of the time they will hang out in circulation doing nothing. Okay, they're monitoring, they're looking for stuff that shouldn't be there. When you scrape yourself on your skin, neutrophils and monocytes can move out of the circulation and into the tissues to combat whatever infection you have. So yes, they, like neutrophils and monocytes, are called phagocytic sites. I don't want to blow everybody's minds here, but if you were to put a sperm cell in the circulation for whatever reason, yes, it would probably be tagged with antibodies and be tagged for destruction. But chances are, now this could happen too. Like let's say that you have like some kind of infection already, like I don't want to say like a urinary tract infection, but more like a yeast infection, or what we call vaginosis, which is real. That is Gardnerella It's Gardnerella. It's the type of bacterial species that causes vaginosis. It's like Gardnerella vaginosi. It's something Latin. Don't quote me on that, but I know it's Gardnerella, and it'll, so you're already going to have a white response in the vaginal canal. You're going to have a white blood cell response in the vaginal canal. If you were, yes, yeah, and I think they diagnose it even through like an odor. It gives off an odor sometimes, from what I hear, and they just know that it's Gardnerella. Yeah, okay, I thought so. I wasn't, obviously I wasn't sure. I get wet preps in when they do a pelvic inspection all the time. Most of the time it's the, it's like the advent of these Tinder apps where some of these people just are they don't think ahead on some things, and then they afraid they've been exposed to some STD, and they come into the ER and they want to be tested for herpes and God knows what. They'll do these pelvic exams. So I get a lot of those in the middle of the night on the weekends. So yeah, sometimes it's and sometimes barring how the epithelial cells look, because Gardnerella will feast on the epithelial cells and use that as energy. We call those clue cells. So if I have a positive clue cell with a white blood cell response in the pelvic pain or in the pelvic area or from the wet prep, then chances are, yes, you're going to have a white blood cell response, and it could respond to the sperm. Sperm are huge. Sperm are big, and they're probably hard to take down. But that's kind of what white blood cells can do. They can get together and tackle something large. I mean, the body can get rid of parasitic infections. Eosinophils do it, and parasites are huge. They're big worms. You can see them. You see them with your naked eye. And the fact that they can live in your small intestine, your body will eventually be able to flush it out, um, and rid itself of a parasite. And it does this using the white blood cells, which is kind of cool. It makes it so it's hard to attach. It makes it so it's hard to attach to the GI lining. It creates a bunch of mucus and it kind of like slides it out. I don't want to say like a laxative, but it's kind of like the same concept. So okay, well, I think the next one is Lab 3, which is a whole another ball game. That's going to be a much bigger lab to go over. We've I'm glad we kind of got some questions going on and some comments. It really helps this hour just run by fast. So I thank you guys for that. Sundays are always very active. I love it. I wish I could get that on my 8 a.m.s and on Wednesdays and Fridays. But thank you guys. Do you have any other questions for me? All right. Oh yeah, that's right, Kaylin. See, I'm glad you said that. All right, hold on. Let me find it. I got to get my email. The link. Yeah, that is important. I almost forgot. All right, wrong. Let me see here. I just heard that chat beep. Hold on, let me find this. All right. All right, here it is. I'm gonna wait. I'm gonna wait for you guys to fill that out, and I'm gonna stand by if you have questions. So if it's got like a title or name, I think you can just put like Mattmiller Sundays 1/24/21. The date, Sunday evening with Mr. Mattmiller or Drew or whatever. And I'm gonna hang out, and you guys let me know when you're done. I want to make sure it works for everybody, and then you're free to leave. I think they do, Lakeisha. I think they do. I've had a lot of questions on that. I've sent out emails, but I can't get anybody to respond to me. But I'm here to cover both lab and lecture material. So I'm going to assume that yes, it does. But I encourage you guys to come back a lot because you're going to get hints on things. Yeah, neither can I. We're rolling. This is something new. So I think we're all kind of figuring out as we go. Just yeah, Africa. Just put Drew Sunday evening, 12421. I think they just want to know which one you attended. Okay, see you later, Caitlin. All right, Terry. Yeah, we'll catch you Wednesday. We'll do some hands-on stuff if you come. If not, hang out with the Zoom. All right, Karen. See you later. Good luck on your quiz. All right. I'll eventually try. I think I'll figure out. I want to work on your name. I'm terrible at names. Some Hannah, perhaps. The J scares me, but I think it may be silent. Samantha maybe. I'm not sure. I'm not gonna embarrass myself. Okay, some Jana. Okay, some Jana. All right. Thank you for that. People always think that my first name's Matt, so they call me Matt, and my whole last name's actually Mattmiller. It's hilarious. Cool. Some Jana. I'll remember that. If I see you later on down the semester, I'll remember that. See you later. You too. All right, Africa. Enjoy the rest of your week. We'll see you next time. All right. See you, Lakeisha. I think we're all good. All right. See you guys next time. Good luck on your exams or quizzes.